All right. So, well, thank you very much for the invitation especially to Marty and Paul for inviting me to collaborate in this set of studies over the years. I'll be presenting actually a little bit what uh, how Carly already presented in a slide. I'm going to present like a few minutes of that study she talked a little bit about that we did with Flora Pompano and the impact of salinity on immune function or immune response and also fatty acid production. So as she said, we're still not published this work, but we hope we can get it out very soon. So obviously, this is all well known to all the audience. We want to be able to figure out ways to raise marine fish under low salinity levels. And what I mean by that is less than 10 ppt, but obviously could go even lower than 5. With the idea of decreasing chronic stress under hyposalinity. Because this could lead to a lot of problems, including increased susceptibility to pathogens. And Vibrio is a very common group of bacteria that is very prevalent, right, and associated with a lot of aquaculture health issues. So we thought that would be a good one to target in this study. And besides that, we found very little to no studies looking at the potential stress of low salinity or hyposalinity conditions in relation to immune function or pathogen susceptibility in this Florida Pompano. So our objective was to assess how did the species respond to the presence of this pathogen when reared under low salinity. And we hypothesized that these fish would, and we've also, I'll mention showing my next slide, these are our juveniles, that if we held those fish under low salinity, 10 ppt or lower, that we would see higher rates of Vibrio infection in these fish. And on top of that, we would also see changes in transcript related to immune function and of course, osmoregulation. So this is a schematic of the experimental design. The fish were provided by our collaborators, Pro Aquatics, and they were juvenile, about 5-gram mixed sexes. We had 5 different salinity, 4, ranging from close to marine or marine all the way down to 8 ppt, hyposmotic. We exposed the fish under these conditions for 14 days, and we cultured, like Carly was saying in the previous presentation, she developed a method, PCR, but also there was some culture going on. So that's day 0 and also day 14. We also, at both time periods, we measured the fish and imaged them for taking notes on the potential lesions. And then we collected spleen, liver, and gills for transcriptomics and fatty acid analysis. So in like most of the data that I'm going to show, actually all of it is negative data, meaning we did not really see much under these conditions, both in terms of, presence of lesions associated with the Vibrio that was very obviously present in the systems that we studied, but we did not see that. There was no difference in any of the external or even internal signs. When we looked at the data on growth, which is, growth, quote, very limited, right? Only 14 days. But anyways, you can see that the fish maintained at the higher salinities grew less than everybody else, even the fish held at 8 parts per thousand. So again, which shows that the fish were doing relatively well under these conditions. This is the principal component analysis data showing the transcriptome results. And the salinities are shown in different colors. So 8 would be blue, right? And 8 parts per thousand would be blue. And then we had data at 0 and data at 14 days. And those are shown in circles and triangles. So the thing about this graph that shows basically that there's really no clear distinguish or separation or clustering going on here that relates to salinity. That's the first thing you see. The second thing is that there is clustering, but that's mostly driven by the time in the experiment. So day 14 fish drifted away basically over time from day 0. So we did not see any significant effects on differential gene expression nor in relation to low salinity, and it was mostly driven by time of sampling. There was a couple of genes, and Carly mentioned those, where we did see some sort of trend going on, but besides those, there was really nothing else happening. When we looked at the fatty acid data, which is shown here, basically it's also a way to clustering the data, looking at the way it clusters based on date of collection. So day 0 would be the circles and the asterisks would be day 14. You see again, no clustering. There was no clustering related to salinity. Most of it, again, changes over time, what you would expect, because the animals are putting in a lot of somatic growth during this time, right? So there should be a lot of changes happening. There's also probably uptake of fatty acids through the diet that are being reflected here at this point, day 14. And those that are shown, those fatty acids that are shown on the slide there are the ones that drove most of the clustering there, and they're mostly long chain PUFAs and some of them. And I guess we were not that surprised to see that, but we were expecting more changes related to salinity, which we didn't see. So in conclusion, we observed that low salinity and abundance of Vibrio in these systems did not impact fish growth rates, osmoregulatory or immune-related gene expression, or fatty acid biosynthesis. And overall, these results would suggest that these Florida Pompano juvenile fish are sort of resilient to salinity changes, or being reared these hyposalinity conditions, at least under the length of exposure that we tested, which was 14 days. It would be interesting to see whether the similar patterns are maintained if there's much longer lengths of exposure. And then there's another pending study that was finished by Harvard Branch last fall, where they tested different diets, so methionine analog, zinc, and methionine proteinate, and zinc alone at 11 parts per thousand. And they also tested methionine alone, which would be the control group, under 30 parts per thousand, and collected samples, livers, and muscle, days 0, 1, 3, 5, and 7, for transcriptome analysis. So we have a ton of data that was sequenced by Novogene over the last few months, and we are basically holding on to that data until we figure out how to analyze it, because we don't have anybody doing that right now. But anyways, that's sort of the last slide I think I had. So I hope I didn't take more time than I needed, but if there are any questions, I'll be happy to answer them.
Thank you very much, Marisol. We're going to hold off on questions until all of the subcontractors have presented, to try to stay on schedule, but we can take the first part of our roundtable discussion to bring up any questions that come up. So please jot them down.